Welcome to the explanatory video for our Direct PCR genotyping kits, the easiest and fastest solution for genotyping without DNA extraction. Before we begin, let's set up your PCR cycler and ensure it's ready to run. The protocol consists of two simple steps. A two-step PCR cycle, 5 seconds at 95 degrees Celsius followed by 30 seconds at 60 degrees Celsius, repeated for 50 cycles. A melting curve analysis, measured from 45 degrees Celsius to 85 degrees Celsius. Make sure fluorescence is recorded during the melting curve measurement in the FAM channel. Fluorescence measurement during cycling is not required, but it is essential during the melting phase for proper data analysis, as you will see later. One of the great advantages of this protocol is its universal compatibility with all our genotyping kits. For this demonstration, we will use blood samples as our primary sample type. To prepare a blood sample, pipette 5 microliters of blood into 495 microliters of PCR-grade water to dilute it. This quick dilution eliminates the need for complex DNA extraction, saving you valuable time. If you're working with other sample types, such as DNA extracts, saliva, swabs, dried blood spots, etc., please refer to the user manual or contact us for specific instructions. Now, prepare the PCR reaction as follows. Take 10 microliters of the diluted sample. Add 4 microliters of the provided genotyping PCR master mix. This is the blue capped tube. Add 6 microliters of the target-specific primers and probes mix. This is the green cap tube. Mix the components thoroughly and transfer the mixture into the designated PCR reaction tubes. Finally, place the PCR reaction tubes into the real-time PCR cycler. Start the pre-programmed protocol as described before. Now, the PCR process will run automatically. One run will take approximately about 1 hour depending on your PCR cycler's fluorescence measurement times and heating and cooling rates. After completion of the PCR run, analyze the first derivatives of your melting curves. Here you can see example data from 2 different genotypes. One sample curve, in green, shows a single peak at 66 degrees Celsius, which is a clear indication of a homozygous wild-type genotype. The other curve, in blue, shows 2 peaks with 1 peak at the same temperature as the wild-type sample, 66 degrees Celsius, and the other peak at a colder temperature at 54 degrees Celsius. These double peak curves are indicators for heterozygous samples which carry both alleles, one wild-type and one mutated allele. By the way, a single peak curve at the colder temperature would be a clear indication of a homozygous mutant sample, respectively. It is really that easy. For more information, feel free to refer to the manual or reach out to us directly by phone or email. Are you ready to simplify your lab workflow? Try our Direct PCR genotyping kits today. Contact us now to request a first test kit. Thank you for watching, and best of luck with your future analyses.